I'm just going to get this because I changed my signal. Here we are. Like last time, we're going to do multi-group data. Even today, we're going to take another step. So today, very much is a recap of what we did last time, with a little extra twist. But a very important extra twist that can make us go from 1 to 2. And when we can move from 1 to 2 way, we can also move to multi-way structures in the future. So let's begin again with these TV data example that I mentioned last time. I mentioned that we do work. I showed you a piece of output from one of our research projects where we work with such analysis of variance techniques. We further develop these techniques and we make them available, and make them available in an improved way compared to what has been around out there so far. So it's part of our research to actually improve these methods and make industry and public sector authority use them to a larger extent than what is done by now. Bang & Olufsen has been part of these research projects, and as I've shown you, we do have, now I changed to this as being really small there, right? Anyway, maybe you can read, it says a two-way Anova up in the corner there, because we produced or made out this two-way Anova that I'm going to share with you as a part of the methodology that we make available with people. Also, and that's where we will go now, because we're not going to use this software. That's for the industry people. We are using R. By the way, the software that I showed you also uses R, so as a machine underneath. I'm going to show you that these Bang & Olufsen TV data, they are out there for you to play around with as examples. I am making a subset now, and in a sec I'll go to R and do it. Just make sure, like that. I'm making a subset where I pick up only one, because it was also done several times, so I pick up one repetition, and I only look at one picture stimuli. So I'm just doing a sort of comparing the three different TV sets with a given picture stimuli. Then instead of what's going on? Here I am. By the way, just to also, I think I mentioned this, but this is again very research close tools topic I'm sharing with you. It is a very good point actually, that there is a very short distance here. I mentioned that in a message also, between the very standard techniques that I'm teaching you in this course and techniques that are actually used out in the industry, various places. So it's not a far distance for you to get up and running and be useful out there here. Let's see. And we made some developments within this field of research, analysis of variance, that we share with the world through this package. You can download packages. You know, this one was made by us, actually a PhD project that was finished just 2 months ago here with me. And this one is a nice one, being already used pretty much globally. So that's nice to see that people use what you produce in research projects Partly, this project was partly sponsored by Bang & Olufsen, but also by other companies, Arla Foods and other Danish food companies. Here's the data set. That's the type of data that we will be looking at today. Here is how it looks typically in the R data frame, in the R setting. We have a Y. We measured, in this case scored, the sharpness of the picture of these 3 different TV sets. So that's the treatment factor, right? Coding for the 3 different TV sets. That's a coding, TV1, TV2, TV3. Then in addition, I also have some persons actually doing the scoring. So there are 8 persons in this so-called sensory panel, or a vision panel in this case at Bang & Olufsen. And each of them scores all 3 TV sets, right? So it's the same 8 persons that scores the 3 TV sets. That is what is different compared to last week. Last week, our minds, we would have 8 patients in one group, another 8 patients in the second group, and another 8 patients in the third group, giving us 24 persons. That would be the setting that goes with one-way Anova that we did last time. Now the setting is different. And just to emphasize that, I just reordered just to show you in this table way, and it's not something you really need to use, but it's just to illustrate. Now, I took the same data and collected in the two-way table that gives the name to the procedure, calling it a two-way analysis of variance, because it's really just this table of data. For each combination of person and TV, I have one observation, right? Each person scored each TV exactly once. So I have a two-way table of data, right? That's why we call it two-way analysis of variance. We are going to do the following. We are going to plot. Why don't I just make sure that I plot both of them? These are the 2 plots we can make, sort of just in the beginning, kind of explorative analysis. That would be nice plots. I plot the box plots for the TV sets and for the persons, for the assessors. That tells me basically the story about what's going on. All I need to do now is just to put some stats to be sure on what my conclusions and decisions will be. TV set 3 has the highest sharpness for sure, right? The other 2 seems pretty close, with TV2 being the smallest one. I mean, I'm not going to change that in all my stats, right? Remember that. All this analysis of variance business, even though you might find it complicated, I'm just looking at the means and making conclusions, right? That's basically what I do. It's not rocket science. There are these 3 means telling the story about TV sets. That's the purpose of everything. Remember that. The purpose of analysis of variance is comparing the means, funny enough, right? And if I want, I could compare the assessors also. And here's a typical set of assessors in industrial use of such human perceptual measurements in your product development or product testing, used by most companies globally. This guy, Assessor 8, is a happy guy when it comes to sharpness. Everything is sharp in the eyes of Assessor 8, right? Assessor 5, well, on average, things appear much less sharp on this guy. Maybe he needs to have his eyes checked. You don't know. It tells the story. We just need now the statistics to tell us, are these differences that we see here, are they just pure random, right? Or do they actually provide me some relevant information that I can use in my future life starting tomorrow after having looked at these data? That's the purpose of the analysis of variance, to make the distinction between randomness and systematic signals that tells us something interesting about the world that we are testing in. For Bang & Olufsen, it's about which TV set should I use for my further product development, or if it's the last stage, which one should I use to put into production, right? And here it comes. Basically, I mean, I could just show you this line and then we were done. Basically, as you remember, the way we moved from single linear regression to multiple linear regression was by just adding another x in the formula statement. And from an R perspective, that's what we do now. We just add both assessor and TV set. Last week, we would only have TV set there, just the treatment factor, right? Now I put both of them there, and it works as it should. Let's just have a look before we look at the results. Here we go. Basically, it's the same type of Anova table as last time with one more row, right? Where we can read off, are the TV sets significantly different from each other here? That's the core question when you do such a test. You're interested in the TV sets. And if I tell you, I mean, you read off things here just as you were taught last time. So, are the TV sets significantly different here? Yes, they are, right? Because we strongly, surely reject the null of them being exactly equal on sharpness. So yes, as we also expected from the box plot, of course, there are some differences. We don't see exactly which yet. That's the post hoc. We'll get back to that. But at least we see, yes, they are different. And then the guy running this panel, for instance, research director Søren Beck, Bang and Olufsen, nice collaborator of mine, professor at Aalborg University. When he is going to control and manage his panel, he would also sometimes maybe check, do my assessors, do my panelists use the scale exactly the same way? No, they don't. So also the differences we see that I mentioned, at least number 8 seems to be the most different one in a way. These are more, I mean, they are more different that could be explained by chance. That's what we see. That's the p-values that express. Could those differences in the means that I see in my box plots, could they be attributable to just chance? What is the probability of seeing such difference in means, given that there was nothing going on? Well, these probabilities are really small here. So something is going on, right? There is a signal in both set of means. They are more different than they could be otherwise, right? If you follow me here, we're almost done. And it's just extra for the remainder of this lecture. That's basically it. See you in a few years. No, sorry. Wishful thinking. Here we are. That's the data. What it all boils down to is looking at mean 1, mean 2, mean 3. I have a double index because now I'm talking about the means of TV sets. I could also talk about means of persons, right? And then I could talk about, whoa. Sometimes we use double mean or just Y bar like that, double bar. The mean of everything. Basically, these means, and these were the means I was looking at before, those are the interesting ones. And we are going to do some stats on these means and trying to figure out what are the differences. Now, sometimes then we go down to this very basic data set of the same structure, where that's coming from the book, from the notes, where we now talk about the 3 different groups. These correspond to the TV sets. Generically, we call that treatments. That's a generic name for that. The focus of an experiment. We call it treatments. And then the generic name of the persons. They could be these blocks. They could be persons, but they could be animals. They could be other things, actually. but the generic name are blocks. So we have 8 blocks of data in our TV experiment. And we discussed the one-way versus two-way setting. We have some names when we go to the design thinking here. We talk about a randomized block design when we talk about a design like that. If you were the manager and you were to design a study, maybe you would have the decision to choose between either doing a blocked experiment using the same people in the different treatment groups, or do what we did last time, make the completely randomized study. That's the name for that, where you put different people in the different treatment groups, right? That is a choice to be made by the project manager, which could be you in the future, right? What to do if you had the choice. Now, if it's done for you, here you have the data, we know what to do, or at least when we're finished today, we would know. It's pretty much always a good idea to do the randomized block design. At least if you expect the blocks to be important, if you expect people to tell you different things, and in medical studies, medical tests, clinical trials, for instance, you do expect that people react differently to drugs, right? For many different reasons, all back to the fact that we are genetically different. So maybe, just maybe, our bodies systematically react different from person to person to the drug that we get in our body. However, in clinical trials, often it's not possible to do blocked design. What would a blocked design mean? That would mean that the same person first should try one treatment for a while and then test whether it works. And then after that, maybe in a waiting period, should try the other treatment. Well, that's nice when it's possible. But it's not always possible to do such nice experiments, because sometimes you have one shot, right? There's one go. You get the treatment once, either it's okay or it's not okay, right? So the completely randomized experiments are done a lot because you couldn't do otherwise. However, whenever possible, when you do testing at Bang & Olufsen or at Arla Foods, then you can do crossovers, as it's called. Then you can have the same people do the different products or the different groups, and then it's definitely usually a good idea, actually. That was almost the intro. I already said that the big question still, like last time, is to answer whether there is a difference in means. That's the question. Generally, we have assumptions about normality and about variabilities being of the same order, of the same size, all over your data set in a way. However, these things are also pretty robust. So there is a CLT, the central limit theorem, helping us also to making it pretty okay often to do basic ANOVA, also in cases where data are not 100% nicely normally distributed. Where the distinction is challenging, of course. But this is just the R to read in the toy data. I'm not going to dwell on that. We have the Y's, the treatment, the blocks, 3 different columns. And then we can plot it like I plotted the TV data, so let's not spend more seconds on that.